Hello, and thank you for joining the Peach Educational Conference as part of the 2021 Virtual Southeast Regional Fruit and Vegetable Conference. My name is Sarah Scott. I'm an extension agent with Clemson University, and I'll be the moderator for this presentation. Pesticide, CEU, and CCA credits are available for most presentations offered during the live Southeast Regional Conference. Check the pesticide CEU guide for a list of approved presentations and also for participating states. The pesticide CEU guide is located in the Event Resources tab under the Media Player. The guide is also available at the Resources Center located on the main menu of the conference platform. Please note that pesticide credits will only be available for registered attendees and only during the live conference. Credits will not be available for any on-demand viewing. There's a simple three-step process to receive your pesticide CEU and CCA credits. First, go to the audience chat box located on the left side of your screen and type your first name, your last name, and the states for which you are requesting credits. Again, go to the audience chat box located on the left side of your screen and type your first name, last name, and the states for which you are requesting credits. You'll need to do this for every presentation to request CEU credits. The second step is to sign out at the end of every presentation. To sign out, go to the audience chat box and type your first name, last name, and the states for which you are requesting credits. You will be reminded to sign out at the end of the presentation. And the third step is to complete the pesticide CEU registration. You only have to complete the registration one time during the conference. This is not required with every presentation. To access the pesticide CEU registration web link, open the pesticide CEU guide on the Event Resources tab, which is located under the Media Player. The pesticide registration web link is located on the front cover of the guide. This presentation is pre-recorded to reduce any technical difficulties. We will be answering your questions live, however, at the end of the presentation. You can submit a question at any point during the presentation by typing your question in the questions box and pressing send. Don't forget to thank our 2021 conference sponsors and exhibitors by visiting the virtual trade show and featured products pavilion. Lastly, don't forget to join us each morning for coffee chats and each evening for networking. Check the conference agenda for details. And now I would like to introduce our speakers, Dr. Sachin, Rutge, and Dr. Christopher Staske, both with Clemson University. They're going to tell us about developing genome editing, reagents, and fungal management strategies for peach.
Thank you, Sarah, for the introduction. So today I will be giving an overview of genome editing and the promises that this technology holds for the future of crop breeding and agriculture. And within my talk, I'm going to discuss some of the research directions and efforts that are ongoing in my lab that use genome editing to essentially build a better peach and an accelerated route towards developing an armillaria-resistant peach rootstock. Then I'm going to hand over the presentation to my colleague, Dr. Sachin, who's going to talk about the development of genome editing reagents and new gene delivery methods in peach, as well as the use of novel biopesticides to manage pre- and post-harvest peach pathogens. So first, let me start out and talk about what genome editing is. So this is a relatively new technology that was a major discovery within the last decade, and recently has been applied to both crop improvement and improvements in biomedicine. The researchers who made this discovery recently won the Nobel Prize, and really this particular technology holds the promise to change the world in which we live in. So genome editing as you can see in the picture here of a double helix DNA strand and a scientist's hands holding what look to be forceps and basically manipulating a gene within the DNA, is just that. It's the ability to go in and precisely modify DNA bases in genes, and these genes code for traits of interest. So it's a breakthrough tool in technology that allows us to more precisely and faster add traits in the context of crop breeding, or perhaps edit the genome of humans to understand the underlying genetics that are driving heritable diseases and things like that. So it's a tool for scientists to explore and discover how genes function in the cell. And this is a high-level slide, but essentially genome editing holds the power and potential to allow us to create the world in which we live in. In the context of crop breeding and agriculture, it gives us the opportunity to use precision genetics to edit the DNA of our elite breeding germplasm. So, for example, if we're facing a new pathogen, we don't have to wait for many years to use conventional breeding strategies. We can essentially edit the DNA and add a resistance gene as needed. In the context of human biomedicine, genome editing has really enabled a shift in the way in which we consider healthcare. So, for example, we've shifted human healthcare systems from being reactive, where you would go to the doctor sick, to being more proactive, where you would go to the doctor when you're healthy. You would sequence your genome and evaluate your DNA for risk factors. And then you could develop a life plan where you can avoid or even prolong the potential for the onset of certain diseases. And genome editing offers the potential to essentially solve all human hereditary diseases. But this technology is still relatively new and does not go without challenges. And we're going to discuss some of these challenges in the next few slides. So let me shift gears a little bit and talk about conventional breeding and how long it would take to basically introgress a gene of interest. So here's a standard setup where we're trying to introgress a resistance gene from a line that has poor agronomics into a line that has good agronomics but is sensitive. So a breeder would go through and make a cross, develop progeny, and then evaluate these progeny over the course of many breeding cycles, which takes years, to develop an individual that has good agronomics with the resistance gene of interest. So we're still looking at about 10 years, possibly even more, to use conventional breeding. But however, genome editing offers this particular outcome in a much shorter time scale. So genome editing is essentially taking the resistance gene and directly transferring it into the sensitive line with good agronomics. So we can achieve something like this with 1 to 2 cycles using the elite genetics in less than 5 years. So this really holds the potential to accelerate our breeding activities. Now let me shift gears a little bit and talk specifically about peach and some of the activities that are ongoing in my lab. So we're focusing on using genome editing to edit the DNA of our most widely used peach rootstock called Guardian. But one of the problems with peach and genetic modification of peach is that peach is considered a recalcitrant species. And this means that peach does not necessarily respond to typical genome modification techniques. So we had to develop a technique that essentially allows us to avert this problem. And what we're looking at here is how we're transforming peach in my lab. So we're developing a technique called the shoot apex procedure, where we grow peach plantlets in a sterile media. We cut the tips off of these and then we bisect them. And it's during this part right here where we bisect them that we introduce the gene of interest with a bacteria. Alongside the gene of interest is a gene that encodes for a resistance to an antibiotic. So we can use antibiotics in our media to select for cells that are transformed. So here's an example of our procedure where we conduct shoot apex transformations. We cultivate these and culture these at a certain temperature for a certain time. And here's an example of what shows us that we're successful. So these are our control samples where we've got bisected shoot apex peach plantlets on a media with antibiotic. And you can see that they're black and necrotic. And if you look here on the right, you can see that these are our transformed peach plantlets, and they're healthy. There's some necrotic tissue, but for the most part, we've got cells that are actively dividing and living in the presence of this antibiotic, which shows us that they do in fact have a genetic modification. So they have the gene of interest as well as a selectable marker. So we've developed a technique that averts problems that are commonly associated with peach transformation, and we're able to insert genes of interest. So this was a very large milestone for our group. And here's an example of one of our first successes where we've essentially cultured a very healthy plantlet, which is now dividing, and we're multiplying it using in vitro techniques to evaluate it for resistance factors to armillaria. So now let me switch gears a little that we're working with. So in the context of armillaria resistance, there's very little genetic resistance that's available in our germplasm pools. But there is one peach plum hybrid rootstock called MP29 that has been demonstrated to have genetic resistance to armillaria. So in our lab, and colleagues here at Clemson, we've essentially set up an in vitro co-culture system where we've taken these different rootstocks, MP29 here on the bottom and Guardian here on the top, and challenged them with the armillaria pathogen. And when we did this, we looked at genes and what genes are being expressed in MP29 that are not necessarily expressed in Guardian during this infection course. And we found an interesting gene that is very active during the infection. And when we're in the process of characterizing it, we believe that it is a primary factor in endowing resistance to armillaria in MP29. So we've cloned this gene, and we're testing it now into the Guardian genome. Another gene that we're working with is the gene that has saved the American chestnut tree. And in this picture here, you can see these very massive trees. The American chestnut was almost eradicated completely during the late 1800s by a fungal pathogen that came over from Europe called the chestnut blight. And essentially some researchers discovered a gene in wheat that endowed resistance to another fungus. And when it was, its gene product was inserted into the chestnut genome, it essentially caused the chestnut blight pathogen to be unable to create a wood infection. And essentially the pathogen produces something called oxalic acid, and the gene inactivates this oxalic acid. We found that armillaria also produces a similar metabolite. So we're testing the efficacy of the OXO gene in peach roots to see if it has any resistance factors against armillaria. Now, another thing that we're doing that is not necessarily a resistance factor, but more or less trying to develop a tool to enable a much faster paced breeding mechanism for peach. So this is called fast track. And as many of you know, that tree breeding is a very long process. It takes many years for trees to flower. And what we're doing here is we've genetically modified the native peach flowering time gene by turning it on all of the time, which would endow continuous flowering. So this would allow breeders to essentially make crosses year-round, and we could reduce tree breeding cycles, from 7 to 10 years to less than 5. So this is sort of a parallel approach that we're using genome editing to develop a tool for rootstock breeding in peach. And last, I'd like to acknowledge my colleagues and funding sources here. So Dr. Greg Riegert is our rootstock expert here at Clemson University. Dr. Ksenia Gosick is our peach breeder and geneticist. Dr. Guido Schnabel is the pathology expert. Dr. Jeff Adelberg is our tissue culture and micropropagation expert. And I'd also like to acknowledge Chalmers Carr and Titan Farm, as well as the South Carolina Peach Council, for funding this project. Thank you.
Thank you, Sarah and Chris, for building up the platform. I would like to just reiterate few of the things so in peach, there were developments all over, in the plant breeding, where the methods have advanced over the time, gradually, from random mutagenesis to site-directed mutagenesis. And this has happened over the years. And now we call this site-directed mutagenesis procedure as genome editing. So this genome editing is none other than like inducing mutations at a particular site. So the mutations do happen in nature as well, but at a much slower rate. And the breeders were selecting those mutations with a lot of effort. Now the difference is like we can actually induce these mutations much more precisely into a particular gene and then at a particular site in the gene and can select them. So it actually reduces the time, as Chris mentioned, like it reduces the breeding time, and that has an advantage. So the genome editing, actually, it is based on two components. So one component is the genome editor or the enzyme that actually has no specificity, and the another component it has is like the guide RNA, which actually directs this enzyme to a particular site in the genome. And this, there are multiple genome editors known right now, like they are known as CRISPR-associated proteins or Cas proteins, and they have different attributes. So for the peach application, we chose an enzyme called the Cas12a or CPF1. So this enzyme utilizes a guide RNA of 43 nucleotides, and it has two components. So one component is 23 nucleotide long site, like which is known as protospacer, which allow recognition of a site in the genome. And there is another 20 nucleotide long part, which I'm actually pointing with the cursor at the moment, like which actually help to actually bring this enzyme to a particular site, which is known as PAM. So it's kind of help anchor the enzyme to a right site. And this PAM we can compare with like a kind of a parking sign. So this enzyme is parked at this particular site in the genome. And then when it's parked at the right location, it actually induces point mutations. And these point mutations are induced by a process in the cell where the plant cell tries to repair these double-stranded breaks that were actually created in the DNA by this enzyme. And then in this process, induce the point mutations. So the other component of this mechanism of gene editing is like the gene delivery method. So once you have these reagents available, you need to deliver them to the plant cell. And as Chris mentioned, there are multiple ways to do it. So it could be done biolistic approach. Like biolistics is none other than like you shoot the gold particles coated with DNA into the plant cells. You can utilize the viral particles, which are disarmed, so they don't cause any disease, but they can still deliver a cargo in the plant cells. One can also utilize the soil bacterium, which has inherent capability of delivering DNA. And in this process, they deliver these genome editing reagents to the plants. One can also utilize the nanoparticles, which because of their size could actually travel across the plant cell wall, which is just like a wooden case around the cell. So they can actually pass through and deliver a cargo inside the cell. So to deal with the to deliver like these genome editing reagents, we actually selected a trait. Like we thought, like as Chris mentioned, we thought like we could actually try to reduce the duration of breeding. Like, and that is, as has been mentioned earlier, it is fast track breeding. So the plants actually for the flowering, they receive multiple clues from the environment, which is temperature, light. So these clues like are received by the genes known as floral integrators. So these floral integrators receive and integrate these signals and induce transition of the vegetative phase to the floral or the reproductive phase by utilizing some of the genes which actually determine the meristem identity. So they tell the meristem, shall it grow into a vegetative shoot or turn into a flower? So these genes are known from quite some time. So there are two genes. One gene is known as FT or flowering locus T, and the another gene is known as the TFL1 or terminal flower 1. These two are the floral integrators, and they have antagonistic functions. So the FT promote flowering and the TFL1 repress flowering. So one can actually create the reblooming plant or constitutively blooming plant by actually either overexpressing FT, which will be a transgenic approach, or could silence the TFL1. So we decided to choose like a non-transgenic approach by inducing point mutations in the TFL1 gene. So these FT, TFL1, brother of FT, centroradialis, and mother of FT, it's a family of related genes. So we actually align their sequences and find a target site which is unique to the TFL1. So we don't disturb the other genes while only create point mutations in the TFL1. So before actually delivering these reagents to the plant cell, we decided to test these genome editing reagents or the guide RNA for its specificity in test tube. So to do that, like we amplified the target site or a piece of the TFL1 gene using PCR approach. And then after amplification, we added it with the commercially available CPF1 enzyme and a guide RNA that we created and synthesized. And then we actually put the mixture at the 37 degrees Celsius to induce digestion of DNA. These enzymes, if the guide RNA is functional, it will guide the enzyme to the right site and create a point, nick or produce a double-stranded break in the DNA, as has been shown in this cartoon here. So we loaded the product of this digestion reaction onto the agarose gels to resolve these fragments based on their size. And you can see, like, when in a control, when we have not supplied a guide RNA, you will see just a single bend. And when you actually supply it with a guide RNA, when this non-specific nuclease would be guided to a target site, you will see it will produce the digested product, which is smaller than the size of the other products. You will see 3 bands. So that's what we observed. So after seeing this, we decided to actually deliver it. So before delivery, there is another step. One has to develop a expression construct to express these genome editing reagents into the plant cell. So we attached the CPF1 enzyme with the Arabidopsis ubiquitin promoter, and so do we attached the guide RNA module also with the ubiquitin promoter. And the whole cloning process is shown in here. We don't have to go into the details of this. After getting this construct done, we actually delivered it to the embryo of peach plant, and it was derived from the guardian seeds. And then we actually introduced this DNA using the biolistics approach, which I mentioned, like it is gold particles coated with the DNA that we produce, like all these expression cassettes, and we shot that into the plant embryos, and we cultured these embryos into the plantlets. And we tested these plantlets for the mutations at the target site. So we performed the same in vitro digestion assay. And in this assay, if you were seeing like the digestion product, it indicates the mutagenesis has not happened. So in the first few attempts, we did not see, and we are repeating since we have only tried with a small number of plants. So we are repeating this experiment at the moment. So another delivery method, as I mentioned, is we can use the viruses as a carrier of these genome editing reagents. So the virus selection is based on 2 things: the cargo capacity of the viral particle, and the another thing is the host range. So we selected a tomato yellow leaf curl virus, or TYLCV, and this is a disarmed version of the virus. So it does not cause disease, but it can actually deliver the things. So we actually modified this, and then we introduced those expression cassettes for the CPF1 and the guide RNA into the viral genome. And then there are 2 ways that we are using right now to deliver this construct to the plant via the roots. So we were germinating the seed and then dip the roots into the solution containing these viral particles, and then test these plants that come out after the viral infiltration for mutations. And then another method that we are using is dipping the skein into the liquid containing these viral particles and install that into the root stock. And then observe the buds that come out, because with a hope like this viral particle will translate and transcribe and produce these the genome editing reagents into the plant cells, and which will induce mutations at the target site in the buds, and these buds will proliferate into the recurrent blossoming shoots. So the other thing that I want to discuss is the use of RNA interference. So RNA interference is a method that actually exists in all the plants and animals. And here, whenever the RNA is a species, like which is a single-stranded molecule, whenever there is a double-stranded RNA, it actually is recognized by the cell as a foreign entity, and it just gets digested into small pieces. And these small pieces denature and get loaded onto a RISC complex, is a complex of the proteins. And this protein complex was guided by this small piece of RNA to a complementary RNA. So utilizing this mechanism, one can actually introduce a double-stranded RNA, which is complementary to the gene of interest, or a gene into the fungal pathogen like the cytochrome P450, which is involved in ergosterol biosynthesis, or to a factor producing dsRNA like the DCL1 and 2. So we utilize this approach in peach to control the Monlinia fruticola that cause the brown rot, or Armillaria foot rot, or the root rot like that it caused. So we selected this gene cytochrome P451, and we actually measured amount of these dsRNAs we applied on the culture plates, and then co-cultured it with the fungi of interest. And then we tried different doses to find out a dose which can actually reduce the amount. So for this purpose, we use two different methods. We can actually measure the radial fungal growth, and we can also measure the amount of the DNA being produced after application of these. And when we tried the 60 microgram dose, we observed reduction in the fungal proliferation. So it's an indication the dsRNAs could be used for this purpose. Since the dsRNAs, they do degrade in nature, so we decided to actually express them in a bacteria, or express them through a bacteria. So we used a construct known as PL4440, where we actually cloned the piece of the gene which can actually produce the dsRNAs in the bacterial cells, and the bacterial cell after induction can actually secrete it. So we co-cultured these transformed bacteria with the fungi, and then observed that on the plate if they can actually suppress the fungal growth. And indeed, we observed the dose of the bacteria, which is 10 to the power 7 cells, reduced the fungal growth, in some cases very much like the propiconazole, which is a synthetic fungicide. So to deal with the problem of the degradation of dsRNA, the another approach that we took is to take a modified oligonucleotide. So these modified oligonucleotides are decorated, the nucleotides of these oligos are decorated with special sugars that actually provide them stability. So these are known as phenanucleotides. So we get them synthesized for Armillaria malia CYP51 gene, and then we plated that different doses of these oligos onto the plate in a fashion very similar to the antibiotic, and then seeded these plates with the fungal cultures. And then we observed at a dose which is 25 micromoles, we saw repression of the fungal proliferation, very much like the synthetic fungicide. So it gives us a hope one can replace these chemical fungicides with these oligonucleotides, which are more green in nature. With that, I would like to thank my colleagues in Clemson University and also the source of funding. And I will be happy to receive questions and answer them. Thank you.